Okay, students, it is time to begin our notes on meiosis. But before we do, I don't want to start meiosis until we know a couple things about chromosomes. So let's start with chromosome. Let's define what it is, the nature of the chromosome. So it's a long DNA molecule that is supercoiled around histones. And so, really, most of the time, a chromosome looks like this. It looks like I don't know, whatever that shape is. But what I want you to know is that it's like you could unwind it, like like if you were to like take a T-shirt and like take one thread, you could just totally like unthread the whole T-shirt into one long thread, right? And that's kind of what a chromosome is like. It's like, this is the strand, right? And then that strand is super coiled a bajillion times into a condensed package. And that package is called a chromosome. So we've got, hundreds to thousands of genes on each chromosome. Okay. So what does it mean to be diploid? Di means two, ploidy refers to the number of chromosomes. So the definition of diploid is that you have two copies of each. Two copies of each chromosome, and that means that you have homologous chromosomes. So homologous just means that those two chromosomes are the same chromosome number. So for example, you might have like two copies of chromosome 13. You would have two copies of chromosome 2. So this is right here, this is a pair of homologous chromosomes. Okay, so next you may see, in fact you will see, later on you're going to have to analyze karyograms. Now, I don't know if you guys remember karyo and what that means in Greek. I believe it's a Greek root. Karyo means kernel or seed, which is referring to the nucleus. So karyogram is a map of all of the contents of the nucleus, which would be a map of all the chromosomes inside a diploid somatic cell. Typically, that's what we're going to be looking at when we look at a karyogram. So because of that, you're gonna have two sex chromosomes, and you're going to have two copies of each chromosome in this karyogram. So here's what it usually looks like. The chromosomes start, by the way, they're numbered with the longest chromosome is number 1 chromosome, and then the length of these chromosomes gets progressively shorter as you go on. Chromosome number 2 is a little bit shorter than chromosome 1, so on and so forth. And then, dot, 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 all the way up to the 22nd chromosome. Okay. And then what's the 23rd? The 23, you've heard of 23andMe, right? That's referring to the number of chromosome types that we have in humans. It's not the same for every organism. Some organisms have more. In fact, I think all of your pets probably have more chromosomes than you. I can't confirm that for sure, but I think dogs and cats have more than we do. I think strawberries have like way more than we do. They're octoploid, which is really weird. I'm not sure why they are. That means they have 8 copies of every chromosome in their cells. It's kind of weird. But just don't assume that don't assume that more chromosome types means that you're a more advanced species. In fact, I think the fruit fly has more chromosomes than we do. But anyway, when you get to 23, this is where you have the sex chromosomes. So let's look at the majority of the time, right? It's either X, Y, or XX. There are instances when this is not the case. We're going to look at chromosomal disorders later and talk about like how the frequency of that in the human population. But right now, we're just going to assume that it's the majority of cases of XY, XX. Okay, so that's 23. So if you were to count, excuse me, if you were to count up the total number of chromosomes, we'd be looking at 46 total, right? So there's a way that we denote this in genetics, and that is we would say that humans are 2n equals 46. So can anybody guess then, using the process of induction, as to what n would have to represent as a variable? If we're 2n equals 46, what does n represent? You got it? The ploidy, or I'm sorry, no, it doesn't represent the ploidy. It represents the number of types, right? The number of types of chromosomes. We have 23 types. Okay, so, but let's break that down. It means we have 22 autosomes and 1 sex chromosome. Now, an autosome is just what is referred to as a regular chromosome, right? Just doesn't have any sex determining genes on them. So we got 22 autosomes, 1 sex chromosome, that adds up to 23. Got 2 copies of each. We're going to have a total of 46. So this is probably kind of review. You may have forgot some of this though, since you last learned it. Okay, so let's say a little bit more about this homologous chromosome thing. So there's something kind of interesting about homologous chromosomes, and that is that they have the exact same genetic map. Okay, so what I mean by that is that if you were to have 2 copies of chromosome 3, let's say, you would find the same genes in the same place. In other words, their genetic map is identical to each other. They have the same genes in the same locations in the same sequence. So let's write that down. They have the same length and position, location, and sequence of genes. So that's what makes them homologous, right? Is that they're now, this is not to say that they don't have different alleles, because they could have different versions of those genes. And that's where we get this concept of dominant, recessive, right? For example, like maybe this one is dominant and maybe this one is recessive. So it's still the same gene. It's just one might be a different version of the same gene. Also, these genes, by the way, they have names. The Human Genome Project was completed in the 90s, which means that the whole genome has been sequenced, and presumably a name has been given to every gene that we've identified. So this gene up here is called MSH2. This gene is called MSH6. And this gene is called PAX3. And those same genes are located on this other copy as well. So let's just add another note here. They have the same sequence of genes, but exclamation point, they may have different alleles or versions of the same gene. So they might have different versions of the same gene. So in other words, the PAX3 probably has different versions, different alleles of the PAX3. So, what that would look like in Mendel's genetics, remember purple flowers, white flowers, pea plants. That'd be like, maybe this pea plant, this individual is, let's just say it's heterozygous purple. And then this individual over here, so this is a purple flower, okay? Like this plant produces purple flowers, whereas this plant produces white flowers. Well, each of these plants, this plant has, since it's heterozygous, it has a dominant version here, and then it has a recessive version here. Whereas this individual has two copies of the recessive version. Still homologous, we're still looking at the same chromosome, but you can see that there's some variety in there based on that. So, by the way, this is all the result of sexual reproduction, right? Sexual reproduction, the hallmark of it is that it provides a lot of mixing and variation. Okay, questions? Okay, I think we're ready now to talk about meiosis because we need the fundamentals. We need to know, because what we're about to look at is we're about to look at chromosomes and how they move around and all of that. So I want to make sure you know what a chromosome is, and what is on them and all of that. All right, cool. So let's do meiosis. Meiosis. So we're going to describe meiosis right now. I'm going to give you some facts, the purpose of it, and then you can be prepared for a big, massive drawing of meiosis. So it's going to come in two parts and probably take up about 2 pages. So 1 page for the details, 1 page for the drawing. All right, so let's define it. It is the process that forms 4 haploid gametes. Now, a gamete is a sex cell, like a reproductive cell, in sexually reproducing diploid organisms. So I'm kind of qualifying this. There are meiosis occurs in strawberries too. Strawberries are octoploid, but the only examples you're ever going to see is probably diploid organisms, or at least the most common organism that we look at as humans, right? We like to focus on that, and that's what a lot of the activities and questions you get is going to be just diploid. In other words, 1 copy of each chromosome. So if that's the case, then you're going to produce 4 haploid gametes. Okay. All right. So remember that the definition of haploid is that you have 1 copy of each chromosome, and then diploid means you've got 2 of each. And by the way, the cells that are haploid, generally speaking, are gametes, and the cells that are diploid are somatic cells. Somatic means body cells. So, 99.9% of your cells are somatic. But in the reproductive organs, there are some haploid cells. Whoa, what happened? Oh, low power. Charger connected. Okay. Fine. Okay, anyway. Okay, so let's talk about where this occurs. So meiosis occurs in the reproductive organs, specifically in the germ line. So there's a specific layer of cells in the reproductive organs. And by the way, like human reproductive organs are different than plant reproductive organs, obviously, right? Like, and so, but each of them does have a germ line of cells, and this germ line of cells it's a layer that we're going to draw. Okay, so let's go ahead and just assume that this layer of cells here is the germ line, and this layer of cells is specialized to perform meiosis. In other words, and by the way, this is the only place your skin cells are not doing meiosis. They don't know how. I mean, maybe they could, but they're not programmed to do that. They don't have the ability, really. But this special layer here, this germline, what makes it special is that it performs meiosis, a very special kind of division. Why is it special? Because it's a double division. It divides twice, and that's what it takes to get down to haploid. Is you gotta divide, replicate the DNA, divide, and then divide again in order to get down to half, right? So we'll look at that. So here's what you need to know: germline cells, they are diploid or diploid. They are 2N, which means that they do in fact contain 2 copies of every chromosome. But then it's going to divide, and that's called meiosis 1. And then it's going to divide again. And that's meiosis 2. And then what you end up with then is 4 sperm cells or eggs, or depending on which reproductive organ we're looking at. But that's what you end up with is 4 gametes, which are haploid. And so don't forget, you guys, to add N, the letter N inside of these. And what that denotes is that these are 1N, which means that they are haploid. They only have 1 copy of each chromosome. So this is 4 haploid gametes. That's the product of meiosis. Okay, now you might be looking at this and going, "Wait, I don't get it," because 2 divided by 2 is 1, and 1 divided by 2 is 1/2. So if we take 2 and we divide it twice, why don't we get 1/2 N? Well, because we're forgetting that before we divide, we actually multiply. Remember stage S in the cell cycle? Stage S means that we double so we're actually going to go times 2, then divide by 2, then divide by 2. So what's 2 times 2? 4. What's 4 divided by 2? And then what's 2 divided by 2? Back down to 1. So that's how we got to 1N. Okay? Times 2, and then divide by 2 twice. Just want to make sure that makes sense mathematically for y'all. And then remember that, okay. This is where I really want to Okay. There's a question that's going to come up later, and that is that yeah. Yeah, let me take a moment. I want to take a moment here to explain this. Let's look at a scenario where, so this is male, and we're going to say that we're going to make a little note here that the sperm cell does not receive the mitochondria from the parent cell. Okay, why do we care about that? Because the mitochondria actually has DNA that gets passed on. So let's look at what it would be for the female. Because the female does the female egg does receive the mitochondrial DNA. So if we're producing eggs, then you have the end product here are 4 eggs. It only produces 1 egg, but just for intents and purposes, we'll show this. That inside here, yes, it is N, right? You're going to have 1 copy of each chromosome plus the mitochondria. All right. So why do we? So we're going to say that eggs do receive mitochondrial DNA. So what we can conclude, you guys, is that mitochondrial DNA is always inherited from the mother. And this is going to be important later on because you're going to get a question potentially on this Unit 5 test, actually, where you're looking at this pedigree and you're noticing that you're kind of following the chromosomes, and you're noticing that they're being inherited from the mother. And so what you conclude is that, and not from the father. And so what you conclude then is that this must be passed on through mitochondrial DNA because it's never being passed on by the father. It's only being passed on by the mother. And the only way that's possible is if it's carried on in the mitochondrial DNA that is only put inside the eggs. The sperm cells do not deliver mitochondrial DNA because they're too small. They're too small the cell to. It's just part of the process. All right, so that's the side note there. Let's talk now about why meiosis. It's an evolutionary innovation. It's an evolutionary innovation that made sexual reproduction possible. Because before that, if there's no way of having a haploid cell, then you're just going to join the chromosomes will just build up to an insane level. I mean, think about it. Like, what do you get if you join 46 and 46? 46 plus 46, okay, 92. And then you just keep adding that, you'll get an insane number of chromosomes very quickly. So we had to make it haploid. If we're going to join 2 cells together, we need them to be haploid so that when you put them together, you get the normal amount. So this innovation of meiosis, it produces haploid cells for the very first time in all of life's history. And so now it's possible for sexual reproduction to occur. And so what is the result of this? Massive increase in genetic variation. You're getting new combinations of chromosomes that you'd never seen before. And it can happen really quickly too with sexual reproduction. New combinations of alleles. And just to contrast this, the world before this point was all asexual cloning, right? Not as diverse, not much genetic variation. So you can think of this as it's an accelerator of evolution, right? We know that evolution occurs by gradual changes. And if suddenly we have a lot more changes happening because of sexual reproduction, getting new organisms, new combinations, new traits, that's going to accelerate evolution and make a whole lot of new species come into existence much faster than they did before. All right, so that's why meiosis is it's like a adaptive trait, basically, like a really crazy adaptive trait. Because like you can be more adaptive to the environment because you can change faster, right? Bacteria, not so much. The advantage of a bacteria is it can make millions and millions and millions of copies of itself very quickly. All right, I think we're ready for the picture. So I just want to show you guys right now what you're going to produce. First of all, you're going to need two colors. Preferably, if you have red and blue, that's kind of intuitive, right? Or like pink and blue or whatever, right? Because what we're tracing is paternal and maternal chromosomes. And so we need to color code the paternal chromosomes. We need to color code the maternal. You pick the color. I don't care. I'm using the traditional blue and pink. Okay. That's what I'm going to use. You can use any color you want. All right, so here we go. The end product is going to look like this. It's going to take up the whole page. Okay, so be prepared for that. Draw small enough to get something like that. And yeah, okay, everybody got that? Okay, here we go. Remember, this whole thing starts with the germline. Let's start there, okay? Germline. Inside one of these germline cells, okay, by the way, do you guys think that in our little drawing today, do you guys think that we can include all 23 chromosomes in our drawing today? Heck no, right? So I'm going to pretend for a moment, okay, just for simplicity's sake, we're going to pretend that 2n equals 4. We're going to pretend that there's two types of chromosomes, a large and a small. And that's it. So that's all we can fit inside our drawing. But I just need you to remember the whole time we're drawing this, I want you to remind yourself, okay, this is not accurate. There's 21 more other ones and that we're not including. And so let your mind be kind of blown by that, be like, wow, man, I can't imagine drawing this for all 23. I can't even believe that the cell can do that, So just keep that in your mind, that this is a major oversimplification, but that's all we can do right now. Okay, so inside this cell is a maternal, I'll use pink, a maternal large chromosome and a maternal small chromosome. And there is a paternal large chromosome and a paternal small chromosome. Why? Because we inherit a copy from mother and from the mother and the father. You get one of each. So assuming that that's what we have to work with, the very next step in making gametes is that we're going to duplicate the DNA. So we're going to go through a process of DNA replication. And I want to show you that there are two possible ways that these chromosomes can line themselves up. So that's why I'm drawing two arrows, because there's two possibilities. But and you can only go down one road at a time. It's not you can't do both simultaneously. So you're either going to go left or you're going to go right. But anyway, the point is that DNA replication will make it's going to make two copies of. It's going to double the chromosomes, right? So what we're going to end up with is something like this, where you've got two copies of the maternal and two copies of the maternal small chromosome. And, of course, now we've got two copies of the paternal, large and small. But notice the difference. Okay, so I told you there's two possibilities. The difference is in the arrangement. And we call this random orientation. Random orientation. So in other words, there's a 50-50 chance that you're going to get that arrangement. There's a 50% chance you're going to get the other arrangement. Now, this is also sometimes, I think your reading will refer to it as independent assortment. So it depends who you ask. Some textbooks call it random orientation. Your reading is going to call it independent assortment. But I just want to point out, already you might be forgetting that this is not the full amount of chromosomes. So if we have n equals 2, there are two possible orientations. As you've seen, there's the left and the right. How many arrangements, how many possible arrangements would there be if we were to include all 23? Anybody got a guess? What if we did that as an activity tomorrow? I said, all right, students, tomorrow's activity, I'm going to give you some, I don't know, like pieces of candy or something. I don't know, like something to represent chromosomes. And today I want you to, on your desk, map out all the possible different arrangements that we could do. If you had all 23. Could you do that activity in a class period? We get thinking. How about if we just had the whole semester to do it? Think could we do it? Okay, what if I gave you a whole year? What about a lifetime? Maybe in your lifetime, probably not. Because there's 8 million combinations. So however long it would take you to lay out 8 million combinations, yeah, you're not even going to do it in your lifetime. But anyway, I say this because I want to illustrate the power of meiosis. The power of meiosis in creating virtually an unlimited number of combinations. So we would literally have to draw 8 million other of these options, and we're not doing that. We're going to keep it simple at just 2 chromosomes. Okay, so back to this 2 chromosome system. What's going to happen next is something called crossing over. So there's a little bit of overlap. So you're going to have I want you to draw it kind of like this, where you got this this inside chromosome is kind of doing that. And then draw the blue ones where they're kind of doing the same thing, where they're kind of crisscrossing a little bit with the pink. Okay, yeah, that's visible. And yeah, and then do the same thing over here for this possibility. Which would be like this. Excellent. Okay, we got it done. That's called crossing over, and it produces even more variation. I mean, there's going to be so much variation here. Like, it's going to be out of control. So much variation. And don't, it doesn't stop there. There's going to be more later on. I'll show you. There's even more variation later. But the point is that, you know, there cannot be another you ever. The odds are it's impossible. The only way there could be another you is if there's identical twins. In which case, but even then, yes, you are identical with respect to which chromosomes you have, but you are not identical with respect to how those chromosomes are used. And that's why identical twins can actually become quite different from each other if they're raised in separate environments. For example, there was a case study that was done once on space. What's that? Is that the one in space? Oh, I didn't hear about that. What's that? Brothers, one of them stayed on Earth, and then one of them went up to the ISS. Oh. And after like 3 years up there, they compared their DNA and they were totally different. Yeah. So what that means is that's great. Thanks for sharing. What that means is that your DNA can, that's what we call epigenetics. And by the way, I have a hunch. Okay, this is not. Don't look at me like I know this. What I'm about to tell you. Okay, it's a theory, and I suspect that the current model that we work with, which is the classic, I guess you'd call it Mendelian model, or the neo-Darwinian model using Mendelian genetics. I suspect in your lifetime, it's gonna kind of have a little bit of a revolution. And what I mean is that we're gonna find out later, and this is my prediction. Again, I could be totally wrong. We're going to find out later that there is much more epigenetic variation going on than we know. And basically what all that means is that it's not that Mendel was wrong. It's just that Mendel was right. But also, as Eli pointed out, also during your lifetime, even though the sequences of your genes aren't changing, there are epigenetic modifications onto those genes that change how they are expressed. And then those epigenetic mechanisms, get passed on to your offspring. So in other words, if that twin brother in space were to have children, his kids would inherit different epigenetic modifications than the other twin brother would pass on to his kids. You see? So that's what epigenetics is. And so more and more evidence is coming out that that's happening more than we once thought. It's really interesting. And biology, by the way, of all the disciplines, so you guys, I don't know if you knew this, but like Physics, chemistry, biology. Biology is the one that is the most mysterious. It's the one we know by far the least about. And sometimes we as students, we have this shiny textbook that's just all perfect, and we're like, as if it like came down from heaven. It's like this is it. Keep in mind, okay, and this is, I'm not trying to get you to cast doubt on what we know, because what we know is, the evidence supports it. It's just that it's the current model, right? It's the current model, and that model is going to develop and get more detailed as we learn more and more about biology. I think it's cool, especially biology. The models of physics have also changed over time too. Kind of interesting. Okay, why the crossing over? What's the significance? Further increases genetic variation. Even more, right? We're getting even more mixing. Okay, so now anaphase occurs, right? You guys remember anaphase? Anaphase is when the chromosomes separate. So we need to show that. I know this is a really involved drawing, but trust me, it's worth it. It's going to help you guys a lot. So anaphase happens, the chromosomes separate, and so this cell ends up with one completely solid large chromosome, but then it gets like a hybrid one. And I'm going to show you that in just a moment. I just need to get the blue pen. So what it would look like then, right, is something like this, where there's a little piece of it that's blue. And same here, right? And then over here, we're going to have that and that. And there's going to be little pink pieces. And then over here, you're going to have that, and then a little blue piece right there. Is that right? Yeah, that's right. I was kind of second guessing myself on that one. And then this one would have a little blue piece there. This one is solid blue, and then this one's mostly blue. Okay, so now that that's done, anaphase is done. We've separated the chromosomes. Which, by the way, that would be this division here. This first division is called meiosis 1. It's the first division that we have. Meiosis 1. So, yeah, okay. And by the way, every time there's a division, the number of chromosomes divides by 2. All right, because we had 1, 2, 3, 4, 5, 6, 7, 8. And now here we got 4. So 8 divided by 2. So now we're having the chromosome number each time we divide. So next is meiosis 2. We're going to divide again. And that's what makes meiosis unique, is that it divides twice. Meiosis 2. All right, I think you guys can do the rest. I'm gonna draw it, but you're capable of showing me the products. We're gonna go ahead and say that these are sperm cells. By the way, keep in mind that the product of meiosis is not 8 sperm cells. It's 4. So remember, we're not doing both of these simultaneously. We're either doing this or we're doing this, just depending on how they arrange themselves in the beginning, right? So just remember that. I hope that doesn't confuse you guys. The product of meiosis is 4 gametes, not 8. All right, so there's that. The law, and by the way, the law of segregation says that the chromosomes will separate. You'll learn more about that with Mendel. So, in other words, they're not inherited together. Law of segregation says they will separate. All right, so if they do separate, what do we get? Well, there's a lot of possibilities, but let's draw all the possibilities. We could have this and this, or we have this and this, right? Or we could have, right, if you separate these chromosomes, you're going to end up with that and that. Everybody following the logic here? Yeah.
What's the word next to together?
Oh, sorry. Oh, yeah, wow, that is hard to read. Inherited? Yeah. Inherited together, not inherited together. Okay. Anyways, you guys can probably get the rest of this, but I'm going to do it anyways. Okay, so just something I would like to point out here is that every one of these, I know you can't make you make 4, you don't make 8, but all 8 are different, right? There's no repeats. These are all unique, and that's significant. The reason that that's significant is that, again, the genetic variation thing, right? Now, so there's, even in a 2 chromosome organism, there are 8 varieties of sperm cells that could fertilize the egg. 8 possible varieties. How many varieties would there be in a 23 chromosome human being? I can't even do the math, but we know it's going to be at least more than 8 million, because there's 8 million orientations and each orientation has 4. So I guess it's 4 times 8, 24 million different sperm cell variations, I guess. Right? So that's pretty crazy. Okay. Last thing we're going to draw is just a simple depiction. This is really going to be quick and easy. We're pretty much done here. But the last source of variation is called random fertilization. Because now, let's just say that you've got 24 million sperm cell varieties. Let's say, how many egg cell varieties would you have? 24 million, right? So, how many different combinations of sperm and egg cell varieties then do you have? Well, that number is just so unfathomably large that I can't even figure it, because like, right, if this just continued on to 24 million, and so did this, like 24 million rows, and then you tried to go, okay, well, there's this possibility, that sperm with that egg, that sperm with that egg, that sperm and that egg, that sperm and that egg, and then you tried to do it for every possibility down to 24 million. I mean, it's not even like, you can't calculate it hardly. You can calculate it, but the number would have so many zeros that it would probably fill up the whole page. So it's just not possible for there to be like another you, right? Like, even if, like, you know, your parents had another baby, the next baby is not going to just by chance be just like you. It'll always be different. There's an virtually unlimited. It's not unlimited, but it's virtually unlimited number of possible combinations. And this is all meant to illustrate the power of sexual reproduction when it comes to variety, producing variety, right? Variation. Okay, any questions? It's a wrap, so I'll go ahead and